you have questions that you might like to ask, or just totally fine.
Oh, no.
Sometimes you may find that you overthought the question and you interpreted things differently. And this is one reason I think it is good to go back through the exam, because it may trigger the next time you want to ask a question during the class exam, try to clarify something the way that I'm thinking or the scope that I'm looking. You have experience about the way that we grade and the scope of the questions and the style of the exam. I just think it's helpful to sort of see what went well and what did not go so well. So let's keep going on the theme of gamete production. And this time we're going to look a little bit more at the female side of things. And so here's a little diagram of the reproductive tract of females. And this is going to, so this is sort of a cross-section laterally, if you will. And we have vagina, the rectum, and the uterus. And here's the uterus, shown here. And on either side are these two tubes that extend out. And these are called the fallopian tubes or the oviducts. Then we also have the ovaries themselves that would contain the female gametes. And interestingly enough, in mammals, and humans included, the oviduct is not exactly a contiguous structure with the ovary. So there are membranes that sort of wrap these two together in a connected fashion, but it's not an absolute, one direction only possibility. So once in a while you can get eggs that are ovulated that don't make it into the fallopian tube. Most of the time they do, because these little finger-like projections on the end of the fallopian tube are kind of beating and sweeping up any eggs that are ovulated right in the right kind of area, and they'll sweep them right into the oviduct. But I just point out that there's not necessarily a physical connection between those two structures. So the sperm, when they are deposited, they're going to be here in the vagina. They're going to have to make it through the cervix, which is sort of a muscular tissue with a small opening in the middle. They have to make it all the way through uterus, all the way up the fallopian tubes. And usually the oocyte is somewhere here in the proximal or the end of the fallopian tube waiting there to be fertilized. So they've got several centimeters that they need to swim in order to find the egg. So oogenesis is our term for the overall process of making female gametes. And once again, we're going to start with the diploid stem cell. It's going to go through several rounds of mitoses. It will make a primary oocyte that's poised to head into meiosis. And in females, humans, this is a point where in the embryo itself, we've already set aside the primordial germ cells, and they're going through these rounds of mitoses. So they do get to the point of making all of those primary oocytes, and then they stop. So they arrest this process until we get to puberty. And then at that point in puberty, these cells are there, they've been waiting, and they're going to wake up, as it were, a few at a time and proceed on through the rest of this cycle. So they will wake up and they will then complete meiosis 1, and you'll get your secondary oocyte. And then they arrest again. So you would think that they would have gone ahead and completed meiosis all the way so that they could at least make a final haploid cell that could be fertilized by a sperm. But they get on the right track and they don't quite complete the very last step. So they start into meiosis 2, but they arrest there. And finally, when the sperm comes, at that point, meiosis 2 will complete, the sister chromatids separate. One of those daughter nuclei will be the pronucleus of the female oocyte that fuses with the male sperm pronucleus. And the other one just gets released as a very small cell called a polar body that degenerates. So this is another difference with spermatogenesis and oogenesis, is that in the case of spermatogenesis, as these cells were dividing, from one primary spermatocyte, we eventually got 4 haploid sperm. because we started out with a diploid cell that went through meiosis and made 4 products. In the case of oogenesis, we're only going to get 1 female gamete out of this whole process. And during this process through meiosis, in general, only one of the daughter cells inherits really almost all of the cytoplasm. So in meiosis 1, we have the 2 daughter nuclei, but one daughter gets very little cytoplasm and just degenerates. It's called polar body. The other one gets all of the cytoplasm and survives, goes on to complete meiosis 2, and once again, one of those daughters gets all of the cytoplasm, and the other one gets nothing. So this means that we have one sort of big oocyte, it's haploid, it's been through meiosis, but then the other 3 meiotic products are non-functional. Okay. So you could also kind of compare and contrast and think about when do we start to see structural or functional characteristics occurring for sperm versus egg. And, again, this is a little bit of a matter of interpretation or how you want to call it, but in the case of oogenesis, since we do see this structural difference of inheriting all the cytoplasm or really none, you could argue that that's the first feature of differentiation, and so to say that that starts really early in the process. And so, this would be in the ovary as puberty is proceeding. In the case of sperm, they go through the whole business of meiosis 1 and meiosis 2, and then here during spermiogenesis, when the sperm are taking on their specialized structure and shape, we could say that's when differentiation begins. Okay. So this I already touched upon, is that in fact the male sperm in mammals, when it comes, is going to fertilize the female haploid cell in the secondary oocyte stage. So with this being the case, you could say that mammals never really make a true ovum singular or ova plural. So just because they never quite get to this stage. Now, it's also interesting that that is not exactly always the same for every mammalian species. This is a case for humans, but we'll have a slide later that looks at some different stages that different species fertilize eggs at. Either primary, secondary, or a true ovum. Just depends on the species. So when fertilization occurs, we can track this using dyes for the nuclei. So blue is dappy, it's a common stain that we can use to look at nuclei. And then green is an anti-tubulin antibody, so this is basically immunohistochemistry looking at tubulin. And all cells have lots of tubulin spread all over the place, but once they start forming mitotic spindles, or once you talked about a sperm tail, that can be recognized as more of a linear structure. So here's the male pronucleus, and all those flares are microtubules surrounding that nucleus. They're emanating from the centriole. The female, okay, we see 2 blue spots, and that's because we had this secondary oocyte that needs to complete meiosis 2. So we're seeing the 2 daughter cells come out of meiosis 2. One of them is being kicked out to be the polar body, and the other one's going to stick around to become the female pronucleus. So then male and female pronucleus can migrate together, they can somehow find each other and fuse, and probably this has to do with the microtubules coming out of that centriole. And then they're going to set up their first mitotic spindle. So here are microtubules arranged in a spindle formation across the DNA. And just to point out that the sperm cytoplasm, such as it was, the sperm mitochondria, the sperm centrioles, and the sperm tail, all get encompassed into the interior of this egg cell. So the sperm tail, eventually it's going to degenerate. The centriole persists and becomes part of the mitotic apparatus. The sperm mitochondria all degenerate. So you may have heard of this mitochondrial Eve situation, and that is that for all of us, all the mitochondria that we have in our cells all came from our mothers exclusively. So So even though the male mitochondria might have been part of the cytoplasm here, they somehow don't make it, and they certainly don't replicate and survive and divide. So mitochondria themselves actually have some DNA in them, and so that's always passed down in mammals through the female side of the family. Okay, here's our slide that looks at where is the egg in terms of maturity when fertilization happens for different sorts of species. And for most mammals, it's going to be in the secondary oocyte stage. But, you know, for random cases, it's different. Dogs and foxes, their sperm actually fertilize at a much earlier stage in oogenesis. Now, you still have to go through meiosis and kick out the polar bodies before you finally get fusion of a female and a male pronucleus, but the timing of this is just earlier in development. Then we seem to see a lot of the invertebrate organisms also fertilizing at the primary oocyte stage. Just a variety of possibilities in how this is done. In some cases, sea urchins, they actually do go ahead and make the true ovum. Okay, so now I'm going to turn to what are the cells that surround the egg in the ovary as it's developing. And these all have some structure to them and some names associated with them. So back in the embryo, our primordial germ cells are busy going through mitoses and making a good healthy population of future germ cells. And they're surrounded by a small group of cells called granulosa cells. So these structured together, oocyte plus granulosa cells, are called the primordial follicle. And in general, follicle is the term that we're using to describe the oocyte plus associated cells within the ovary that you will see as these oocytes undergo maturation over time. So here in the fetus, a number of different primordial follicles, they're kind of in a quiescent state. First they're divided, but they reach a point where they're pretty much done. And then they're arrested there until puberty happens. And at puberty, so roughly around 12 or so years, give or take a couple of years in humans, these follicles start to wake up, as it were, a few at a time. So they start to advance to the stages of further maturation, but not all at the same time, just gradually with a few of them every cycle. So the next stage that they go to is called the primary follicle, and at this stage it's not that different except for these granulosa cells. Initially they're more squamous or flat in shape, and in the primordial follicle they're more cuboidal in shape. So it's just one difference that you can see. Here it shows this better on this slide. OO is oocyte, and then you see these flat cells surrounding the oocyte in the primordial follicle, and they become much more distinct and cuboidal here in the primary follicle. And then also just to point out, in between these follicles, you'll see a number of sort of general connective tissue cells, and these are called thecal cells. And they help to just maintain and nourish the development of follicle cells. Okay, so here's our little graph that looks at how many oocytes does the female have in the developing ovary at what point of her life. So this white stripe here is birth, so everything before that is pre-birth in the fetus at 3 months and at 6 months. And so somewhere around 5 to 6 months, this is the time when we've actually gone through all these hundreds of rounds of mitoses, and we have the maximum number of germ cells present. And on the y-axis here is the number of germ cells in the millions, so we're looking at upwards of 6 million cells in the fetal ovaries at this point in time. And then this drastic drop, so they start dying off. We actually lose the vast majority of oocytes even before birth. And then from that point on, it sort of levels out, and then as these cells are maturing, some of those that are left behind in the ovary are gradually kind of dying off. There's a term called atresia. It's A-T-R. This is describing the cell death of oocytes within the ovary. So a large number of these cells undergo atresia. So this is going to leave about 300 to 600 cells in humans that actually would be ovulated over the lifetime of the female, so from age 12 to, I don't know, 40 or so. And then how many of those actually end up being fertilized would be generally 10 or less. So a large number of cells and not that many progeny in the end. Now, it had kind of been the standard standard lore, standard thinking that females at the time of birth, they already have all of the oocytes that they would ever have for their whole life. Because this whole process of going through mitosis, that was done and in fact even diminishing before birth happened. And so this was somewhat of a limitation because sometimes women would be in their late 30s and they're trying to get pregnant and those eggs have been there, for 35 years and they're not always of the greatest quality by that time. But there was no hope of getting any new ones because we're done with that. We've been there, we've done that long ago. And interestingly, about a couple of years ago, this dogma was challenged, at least a little bit, by this publication of this Nature Medicine paper, where in reproductive age women, so these are adult women who are ovulating, that you could find a very small number of cells that do seem to still be going through mitosis. And one reason that this might be interesting then is that for women that have fertility problems, if we could identify those cells and manipulate them, maybe we could generate a few extra oocytes that are younger and that are of better quality and could actually be fertilized by sperm. So just an interesting development, and I think this kind of thing is good to keep in mind for science because it always seems like we think we have the dogma of this is how it works, and then well, there's an exception. Okay, continuing on from the primary follicle. We'll get to a stage called the secondary follicle, of course. And this is going to be represented by proliferation of the granulosa cells. So these cells that are surrounding that oocyte will continue to replicate and start to form a little bit more robust of a structure within the ovary. And they'll just have multiple layers. And they begin to secrete fluid. And as that fluid collects around the oocyte, you can see this in the ovary, and that structure is called the antrum. So it's just a fluid-filled region within the follicle. And this could provide a couple of functions for the developing cell. First, it could keep it well moistened, so it doesn't get too dried or anything like that. And secondly, it provides a kind of a padding or an insulation around that cell, which may serve to protect it. So those are some possible functions for the antrum. Over time, this antrum is going to get larger and larger, and that is one of the characteristics that we notice in follicles that are reaching more and more of a mature stage. Then also the oocyte here is going to begin to secrete a layer known as the zona pellucida. And pellucida, the word means clear. So you can think of this as a clear layer. It's secreted by the oocyte and it surrounds it outside of the oocyte plasma membrane. And this is going to include glycoproteins, and this will be important in fertilization. So this begins to develop as well. And meanwhile, the antrum is getting larger and larger. So if you look at what this thing looks like at this stage, we can see our primary oocyte that is beginning to wake up here, surrounded by zona pellucida. Then we have granulosa cells. And granulosa cells get a few different names in the literature. This is sometimes called the cumulus oophorus, which means a layer of cells surrounding that oocyte. And then finally, more granulosa cells, and then outside of that, fecal cells. Okay, cumulus oophora, another name for it is the corona radiata, the radiant crown. So the oocyte is surrounded by all of these cells that sort of look like a crown. Another thing that's happening here is that the meiosis is continuing for that oocyte cell. So we're moving from a primary oocyte into being a secondary oocyte once we go through that round of meiosis. And the granulosa cells are proliferating and the antrium is getting larger. So the final stage for this follicle in its most mature form is going to be called the Graafian follicle. And this will have a very, very large antrum. So here's the oocyte, the corona radiata, and then all the rest of this structure, many times the size of the oocyte itself, is all the fluid-filled contents of the antrum. And then more granulosa cells around that here within the ovary. And generally, within one cycle, you may see maybe 2 to 5 of these cells that are progressing to this stage. And one of them will reach this large Graafian follicle stage the first, and this is generally the one that actually will be ovulated. And the others kind of just die back within the ovary. So usually with females, we have 1 or possibly 2 cells ovulated per month. And it actually rotates which ovary does that. So these can get so large that you can see them by ultrasound. So here's an ultrasound looking at an ovary, and these large dark structures are where follicles have gotten very large antrums, and you can actually recognize these. And one of the things that they do if women go to fertility clinics, if they're having trouble being pregnant, is they'll just monitor what's the stage of follicle development within the ovary. And when the time is right, they tell you, and you can go home and enjoy and hopefully become pregnant. Okay, so here's a diagram of this process within the ovary. You can see these different stages. And my only comment here is that the ovary is not arranged in a nice, tidy, sequential fashion. All of these stages are all intermixed over the entire thing. Once we get to the Graafian follicle stage, when ovulation occurs, the oocyte plus the associated granulosa cells that are part of that corona radiata, that whole structure gets expelled from the uterus. So that will be out there then and hopefully swept up by these fimbriae into the fallopian tubes. And then there's part of that follicle structure that remains behind. And it has a continued important role to play to aid fertilization within the animal. So these cells survive. And they begin to secrete progesterone and estrogen. And this body takes on kind of a yellowish appearance, and so it becomes called the corpus luteum, which I have here in the picture. So here's just showing you ovulation right at the moment of occurring, and you can see there the oocyte plus the cumulus cells surrounding it. A lot of this fluid that's coming from the antrum. But then all of this structure remains behind here, all these remaining granulosa cells, and becomes the corpus luteum. So corpus, of course, means body. Luteum means yellow. So it's a yellow body because of a slight yellowish appearance that it has within sections. And this will continue to secrete progesterone and estrogen. And so this then gives a waiting period. Well, the body says, okay, we have an oocyte on the table. Let's wait and see if it gets fertilized. And let's keep the reproductive tract in a state of readiness so that if that thing gets fertilized, it will be able to implant and become a fetus. So these corpus luteum structures will persist after ovulation for about a week or 10 days. And then if nothing happens at that point, the cells begin to die back, and give it a new name. So here's a couple of corpus luteum. They're not particularly yellow because this has been stained, this slide, but see how large they are? And then finally, once these cells start to die off and disappear, what will remain looks just like a little scar tissue within the ovary. And so for women in their late 30s or 40s, if you were to section through the ovary, you would see a great number of these corpus albicans because of all the ovulation events that have happened over the course of their life. So, in a younger woman, maybe 15, you might only see a few of these. So, these cells are undergoing apoptosis, which is programmed cell death, and then we get this white-colored body left behind. Any questions on any of this? Okay, for the final slides here, we're going to talk about how we are coordinating all of this within the body. So we have an ovary with ovules at different, or follicles at different stages here. We have a uterus, which is undergoing a menstrual cycle and needs to be in a certain state of readiness if that oocyte gets fertilized and wants to begin to make a placenta. We have a brain, and especially the pituitary glands that are making some hormones that help coordinate all this process. So these three things sort of need to be in communication with each other and to assess whether pregnancy has occurred or not. And there's a whole cycle that goes through where everything is dependent on other signals. So let's look at this up here in A. This is looking at the gonadotropins, and these are hormones that are made within the pituitary gland that help coordinate the menstrual cycle. And two in particular are FSH, which is follicle stimulating hormone, which is also written out there, and luteinizing hormone, LH, which is written out as well. And you can see that there's one peak for both of these at one point during the month-long menstrual cycle. And follicle stimulating hormone, you might guess what that is doing. It's basically stimulating the maturation of the follicles. So if you look then in the ovary here at B and see some of these different stages and how they're coordinated, once we get that burst of luteinizing hormone and follicle stimulating hormone, that is stimulating the ovulation of the mature grafting follicle that's growing up in the ovary at that time. So that coordinates ovulation with the hormone cycle. And then we have formation of the corpus luteum, which is going to persist here for 7 to 10 days, and then finally, if there's no pregnancy, it will die back. Okay, what about the uterus? So the uterus, you can divide the menstrual cycle into a couple of different phases, the follicular phase and the luteal phase. And this red bar here is representing the lining of the uterus, which becomes very elaborate over the course of the monthly cycle. So it becomes there's like a proliferative layer with lots of blood flow and influx, and then this will eventually be shed, and you'll lose those cells in that blood over about 4 or 5 days during the menstruation period. Okay, so this is going to be gradually building up over the follicular phase, and then in the next 10 or so days, it'll be maintained there as a nice, rich place for the fertilized egg, if it is fertilized, to land and integrate with this tissue. And then, if nothing happens, it dies back. Finally then, what about in the ovary themselves? The ovary is also making a couple of important hormones, and these include estrogen and progesterone. And you can see how they cycle here. So once we get to the corpus luteum, you see high levels of progesterone and estrogen, in part because that's the structure that's making these hormones. And then those levels will drop when the corpus luteum starts to fade away. So everything's kind of tied together. Oops, I think I jumped to the wrong one. Okay. So the luteal phase, is the second half. So if pregnancy occurs, we basically want to stop this cycle for the next 9 months so that we can have a chance for the embryo to implant into this uterine wall and start making a placenta. So during that time, you really don't want to continue ovulation, and you don't want to have a second fertilization event or a second embryo to come along while there's already a current fetus within the womb. So if fertilization occurs, so you'll see these sorts of changes. The corpus luteum is going to continue to be alive and maintained, and so it's going to continue making progesterone. And these higher levels of estrogen and progesterone seem to feed back and then repress them out of FSH and LH so that those remain low. And with those low, you're not going to stimulate any additional follicles to be matured or ovulated. So that's how you basically stop ovulating for the term of pregnancy. In addition, these hormones also help to maintain the endometrial layer, because the last thing you want to do is have menstruation occur where you might lose the embryo and its supportive layer through that process. Okay, if no pregnancy occurs, as we've already mentioned, these events all cycle back to the beginning again. Okay, so this represents the path the sperm are going to take from the vagina all the way through the uterus up through the fallopian tube, and usually up there in the end of the fallopian tube is where the fertilization event will occur. So with this being the case, you think about, again, some of the mechanisms that might promote this or challenge this process of fertilization. And I don't know why these turned into white here.